Hi, good afternoon. My name is Susana Aguirre. Today is Monday, September 24th. Today, 2018. Today we're going to go ahead and start with our first experiment, and that would be our biochemical testing for microbial identification. This is the Methyl red VogesProskauer and Catalase testing. So we're going to go ahead and get started. All right, now we're going to go ahead and do the second experiment, which is the biochemical testing for microbial identification. Only this one is the Methyl red VogesProskauer and Catalase testing. So we're going to go ahead and start with exercise 1. And they have asked us to label our MRVP broth. So we're going to go ahead and label that. One of them is going to be E. coli, and the other one is going to be so this one's going to be the E. coli, and this one's going to be the S. epidermis. Or epidermitis. All right. So we're going to go ahead and start with the first. I'm going to grab my active culture for the E. coli, and we're going to go ahead and sterilize the tip. Okay. I'm going to also grab my my MRVP broth. I'm going to go ahead and sterilize that as well. Okay. Now we're going to grab one of the pipettes, and we're going to make sure that there is no alcohol remaining in there. We're going to go into the live culture broth, and we're just going to grab a couple drops here. And it says about 4, so 1, 2, 3, 4. Alrighty, and then we're going to get rid of this in our Clorox bin. Now we're going to go ahead and sterilize this MRVP broth. And we're going to go ahead and sterilize our active culture. Alright, now we're going to go ahead and repeat the same step for our S. epidermis, and then we're going to go ahead and leave this in incubation for the next 48 hours. Okay, so now we're moving forward with our next part. We've left the MRVP broth, both of the E. coli and the S. epidermis. We've left it for 48 hours. Now we have gone and did half and half, or equal parts into both of these test tubes. And now we're going to move forward with the testing here of the methyl red regimen, and it's asking us to do 7 drops here and 7 drops here. We're going to go ahead and start that now. So 1, 2, 3, 4, 5, 6, 7, and we're going to go ahead and do that to the other one as well. Okay, we're doing part 3 of this experiment, and this is the VogesProskauer test. We have already removed the top of this agent A and agent B. It is asking us to use 12 drops of A in both, and then we're doing B, we're doing 4 drops in each. We're leaving it here, and now we're going to observe it and leave it here for the next 30 minutes, and we're going to leave it untouched. We're going to leave it undisturbed for the next 30 minutes, and then we'll observe the color changes, and then we'll upload that to our laboratory report assistant. Okay, after we went ahead and did both the burette solution, we can see clearly that the E. coli was positive and the S. epidermis is negative, and we're going to go ahead and record that in our lab assistant. All right, we're going to go ahead and do exercise 2, which is the Catalase testing. We went ahead and gathered our E. coli plate, and we have made already our slide with the E. coli name on there. So what we're gonna go ahead and do is tap dry one of our loops here, and then we're gonna go ahead, once it's everything is dry, we're gonna go ahead and go into the plate, and we're gonna collect several colonies from this plate. So I'm just gonna dig in here and kind of get as many as I can. Okay. And now it says we're gonna go ahead and bring it over to the slide and try to put as much on there as possible. And I'm just gonna grab a little more because I don't see enough in there. So I'm just gonna scrape it a little bit better. Okay. Okay. There's a little bit in there. Trying to see if I can smear it on there. Okay. We can dispose of this in our Clorox. Now we're going to go ahead and put just a drop, it says 1 drop of hydrogen peroxide onto the sample, and we're going to absorb and see if there's any bubbles. Okay. From what I can see, I don't see any bubbles. So what we're gonna do now is we're gonna take a picture of that, and then we're gonna upload it to our assistant lab form, and then we're gonna go ahead and move forward and do the same for the S epidermis. So for now, we're gonna take a picture, do the same steps that we would do here, and we're gonna do it to the S epidermis. Thank you.